Thank you so much for having me. I'm very excited to be telling you about the work that I've been working on in the dark genome, targeting line-1 as a way to activate innate immunity in cancer. So this really illustrates the problem that we've faced. We have been focusing all of our therapeutics on 2% of the genome, the coding part of the genome. That leaves us 98% of the genome, the dark genome, as an opportunity for therapeutic development. So the big question is, well, what do we know about this dark genome? Well, my lab over the last 15 years, we've been focused on understanding repetitive elements. And it turns out that repetitive elements make up 59% of the genome, so the vast majority of the dark genome. And so what have we learned about what these repeats are doing? Well, we've learned early on in my initial work that these repetitive RNAs are expressed in the vast majority of cancers. All the major solid tumors in adults express repetitive elements at very high levels and at the very beginning of tumorigenesis. Others have now found that these things are dysregulated in neurodegenerative disorders. We have now found that these things are activated in chronic viral infections. And finally, we have even seen this to be activated in aging and senescence itself. So this large part of our genome, the vast majority of our genome, is being activated in many disease states, and this is an opportunity for us not only to impact cancer, but also all these other types of diseases, including aging itself. So what do we know in cancer? Well, over the last 15 years, my lab has really focused on understanding what these repeat RNAs are doing in cancer. And what we've learned is that they mimic viruses. In fact, what we've learned is that in a cancer immunotherapy trial where we used immune checkpoint inhibitors, we looked at patients with colorectal cancer and pancreas cancers and sequenced their biopsies before immunotherapy. And what we found was the vast majority of patients that had a response to immunotherapy had high levels of these viral-like elements being expressed in their pretreatment biopsies. So they look like viruses. The second thing that we learned was that these things are replicating like viruses. In fact, they look and behave very similar to HIV. And we actually took a drug, 3TC, that we commonly use in HIV and initiated a phase 2 clinical trial in colorectal cancer. In this trial with single agent 3TC, we saw disease stability in about 25% of patients with this reverse transcriptase inhibitor. So we can deploy antiviral treatments actually for cancer. And this led to the founding of Rome Therapeutics. And the last thing that we've most recently discovered is that not only do they look like viruses, do they replicate like viruses, they look like they can infect the microenvironment like a virus. So these things are actually packaged in these things called extracellular vesicles. What are extracellular vesicles? Well, they actually kind of look like viral particles. And these viral-like particles contain a huge amount of these repetitive sequences. And what we found was that they can actually change the fibroblast microenvironment, the immune cells around it, and even the nerves that are in the cancer cells themselves. And so we have the opportunity not only to target the cancer cells themselves by activating innate immunity, but actually affecting the microenvironment around if we can hit this target. And so what's the attractive target? Well, it turns out that the most common retrotransposon in our genome, which makes up 20% of our genome, actually produces a protein. This line-1 protein actually has 100 copies in our genome that produces two proteins, ORF1 and ORF2. And this creates a protein that binds its own RNA, reverse transcribes that RNA into DNA, and then actually can replicate it in the genome and alter the genome of these cancers. And so we've targeted ORF2, which is the reverse transcriptase, but what I'm going to be telling you about is targeting ORF1, which is the ribonucleotide protein, the thing that actually binds that RNA. Can that be a new therapeutic target for this space? And when we looked at line-1 ORF1 in pancreas cancers, we actually see that in the brown stain here, that higher production of this ORF1 protein in pancreas cancers actually correlate with worsened survival. So if we have an opportunity to target this protein, we have an opportunity to improve the outcomes of some of these patients that are suffering from this highly lethal disease. Genetically, we then used short hairpin RNA. We were able to suppress line-1 ORF1, and we see anti-tumor effects in our patient-derived pancreas cancer cell lines. And when we started to understand what was actually happening when we took out ORF1, we actually find that transcriptionally, we see reductions of proliferation and invasive transcriptional profiles, and we see increases of innate immune response when we take out ORF1. Now, when we extend this to in vivo models, we actually see that the tumors grow smaller, they grow less, so targeting ORF1 has clear anti-cancer growth effects. But as you can see in these histology images, that the non-target control, the one shown on the right, is that this effect is actually going to change the entire microenvironment. And so what you see here is that those tumor cells in this monolithic sort of growth as a tumor, we actually see it turning into more of a fibrotic response that you see in these tumors that we took out ORF1. So not only are we having effects on the tumor cells themselves, we're causing cancer cell death, but we're actually creating a microenvironment change. So this leads to an opportunity of targeting ORF1 as a new way of engaging in the immune response in pancreas cancer. Finally, one of the other things that is hot in pancreas cancer is KRAS therapies. KRAS is mutated in over 90% of pancreas cancers, and so KRAS inhibitors have been used. But one of the common things that we see with KRAS inhibitors and other targeted therapies is that there is resistance that quickly emerges from these targeted therapies. And so we did a simple experiment. We took out line 1 ORF1 and these pancreas cancer cell lines, and what we see is that ORF1 loss actually synergizes with KRAS inhibitors and resensitizes these cell lines to KRAS inhibitors. So this not only offers an opportunity as ORF1 as a target, as a single agent, but also a synergistic play with KRAS inhibitors, which are the hottest thing in pancreas cancer today. So in summary, cancer-specific expression of line 1 makes this an attractive target. This is an attractive target because the sort of window of therapeutic window of effect is going to be high because we only see line 1 ORF1 in cancer cells and not other sort of normal cells. This line 1 ORF1 shields repeat RNAs from the innate immune response as if a virus is hiding from the innate immune system. And this suppression of ORF1 actually has effects in both in vitro and in vivo models of pancreas cancer. And so we see this as an opportunity to engage a new dark genome target that we're looking to find new investors. And what I'm calling this new company is Orpheus Therapeutics, and we will target the dark genome. IP has been filed on the line 1 ORF1 as a cancer therapy. The ask is a $10 million seed to start a new co. Our goal is to develop a line 1 ORF1 small molecule inhibitor or a degrader, and to identify proteins that interact with line 1 ORF1 as novel therapeutic targets, that we have a platform going after other dark genome targets.